So welcome everyone, both to those of you who are here and those of you who are online. It's really my pleasure to have a chance to speak with you briefly today. And so I'm going to really build on what my colleagues said, and I'm going to touch on a barcoding campaign that's close to my heart because I've been involved with it quite a bit, the polar barcode of life. You'll notice a polar bear with a nice DNA on it to showcase what we're trying to do in the north. So why might I be interested in the north? So I'm sure you've heard a lot in the news about climate change, and this little schematic shows some temperatures in the Churchill Manitoba area over years. And you can see there's quite a lot of variability from year to year in the temperature, but overall there's a convincing trend that more hot years have been in the very recent decade and in the past 2 decades, and that the overall trend has been towards increasing temperatures. And this pattern is stronger in polar regions than in other regions. But what effect might this have on the organisms that live there and the communities of biological organisms that make up these environments? And so one of the projects I'm intensively involved with, and this involves also a very large network of collaborators, is a targeted project to really study in depth the flora and fauna of one region. And it's the region of Churchill, Manitoba, which I'll highlight here on the coast of Hudson Bay. Now this was selected because it's a very interesting region where the boreal forest gives way to true Arctic tundra. So it's really a site where different types of organisms are meeting, and a site where we might expect to see change, and we might expect to see some of the effects of change earlier than at some other sites. As well, it's a site that has a fabulous field station, and it's a great place to collaborate with other people and a great place as well for field courses. There's a 4th-year field course here at University of Guelph that's gone up a couple of times. And so I find this a fascinating environment to work in. Also, it's been a big challenge for me and a very interesting life experience. I've gotten to do things I never thought I would ever do. Those people who know me well know I'm not necessarily the most spatially gifted person, so it was a challenge for me, for example, to learn to drive a bus, to teach this field course up in the Churchill environment, and also being a long time pretty much vegetarian, I never expected to be a firearms user either, and so part of my training for working up there was learning how to be a bear guard and learning safe firearms usage and so forth. Of course, only as a very, very last resort. We always use vigilance first as our first line of defense against polar bears, because it's very exciting but also a bit scary to work in the polar bear capital of the world. But it's a fabulous environment to work in, and we really love it up there. And so what did we do up there? Well, we were doing kind of the things that Dirk was talking about, surveying biodiversity, because the scientific community, despite many years of study, we really don't have a complete understanding of biodiversity on our planet. So we wanted to take a focal site and really study it in depth, including the terrestrial environment, freshwater environment, and nearshore marine environment. And also, as my colleague Dr. Hanner said, that we have a certain workflow that's followed, and generating barcode data is not willy-nilly. It has to have collection data, a tissue sample, a photograph, and then it's all linked together online, including the sequence data, the GPS data, and so forth. So it's really a digitized form of data that can be then used for a lot of different types of analyses. And it's all grouped together on an online platform, which anyone is welcome to explore, the Barcode of Life Data Systems online database, which combines the geographic information, specimen information, etc. And so out of this campaign, we've been barcoding all sorts of different groups of life. Flies, wasps and their relatives, moths and butterflies, beetles, etc. Barcoding, in some cases, thousands of specimens, depending on the diversity of the group, and finding, in some cases, thousands of species, depending on the nature of the group. But just getting all that barcode data, I think that's really the starting point. And I think this cartoon really captures my view of what it means to just generate the barcode data. So this is an interesting stamp that was used in the UK, and it was published to celebrate the Human Genome Project. And so as you can see, as they're celebrating the completion of the Human Genome Project, all the little pieces of the puzzle are just making one bigger piece, that it's opening new questions, new research avenues, new directions in science. And I think it's the same way with this DNA barcode data. Sequencing these small bits of DNA from lots of different species is really step one. It's opening lots of new avenues for doing science and learning new things about the world. And so in the interest of time, I'm only going to share 3 little vignettes about the kinds of insights that we're starting to gain, my colleagues and I, with this barcoding data, especially with a focus on the Arctic and in freshwater systems. So the first little vignette I'm going to share is from an intensive study in Churchill to do barcoding of the flies. Now, if you've ever been in northern Ontario, the Muskokas region, or so forth, you may think you have a good knowledge of what it is to be swarmed by biting flies, like black flies or mosquitoes. But until you've been to the Arctic, you maybe have not experienced the full extent of the intensity of the population density as well as the diversity of flies. Flies are incredibly diverse and abundant in the north. But one thing I'd like to highlight is that before our big barcoding project, there were fewer than 300 species recorded from the Churchill area. But after the intensive collecting that we did, as well as the barcoding, this almost increased by 10 times. And so this is almost our own backyard in some ways. This is a fairly accessible part of Canada, and we're almost increasing by 10 times the amount of diversity that was known. So what does this mean about the community structure and the future of these environments under climate change? We're only starting to understand that, but with barcoding, that can help us to get a handle on those kinds of questions. So the second little vignette I'm going to share is that barcoding can give us an opportunity to study shifts in biodiversity over time. So this was a study published by Fernandez Triana, a colleague of mine and colleagues. And in this study, they did barcoding of both modern collections of these kinds of wasps, collected very recently, as well as historical collections housed in great museums of Canada, such as the Canadian National Collection. And the black groups were collected only recently. The red ones were only in the museum material, and the blue ones were in both. And even though more research needs to be done into this, it's really showcasing that even on the scale of a few decades, there have been some important shifts in the most common species in those communities. And this really wouldn't have been possible even to document this in these very difficult to study wasp groups. But with the DNA evidence is letting us understand these kinds of shifts in biodiversity composition over time. And then for the third small vignette I'm going to share is a study by my colleague Beth Clare and colleagues studying bat diet. And now she was studying bat diet not by following the bats around or not by dissecting their stomach contents, which would hurt the bats, but by barcoding their waste, their guano, and trying to figure out what are they eating by barcoding their guano. And so it was very interesting. She compared bats being roosting in a farm area versus a forest area and found that they actually had a fairly similar richness in terms of the species richness of their diet. But what was also very interesting is that they found in this little brown bat species is that the maternity season seems to coincide very closely with the emergence of mayflies, which you may know emerge in huge numbers in a particular time in late spring. And as well, it was very interesting that was using the freshwater insects and the information she was gaining about the freshwater insects from the bat guano to draw conclusions about environmental quality, because these freshwater insects as larvae live in water and they have a wide range of pollution tolerances. So it's actually very interesting how all the components of the ecosystem are linked together, and that by studying bat guano, she could understand more about bat ecology, but also more about the freshwater ecosystem and pollution levels in that area. And so it's just that my final slide, I'll just point out that a lot of this research is a broadly collaborative program involving funding agencies as well as researchers from around the world. And I'd like to leave you with this kind of cool picture. In 2010, the International Barcode of Life project was launched, and we had a launch party at the top of the CN Tower, which was pretty exciting. And in honor of the Barcode of Life project, the CN Tower kindly lit themselves up with the barcode of the Canadian beaver. So we thought that was pretty cool that they did that, but also the illumination in those 4 colors just gives a nice simple picture of how, on the surface, how simple DNA data are. It's 4 nucleotides. It's a digital signature, which means it's amenable to broad-scale analysis using modern computing as well as the internet. So I think the possibilities are really exciting for this line of research going forward.
Thank you, Sally. That's great. Give my colleagues a round of applause for their presentation.